Why, hello, everyone. So, we're going to talk about viral characteristics today in part 3. So, what makes a virus? Well, a virus is any microscopic particle that can infect other living organisms. One single virus is called a virion, which lacks a cell membrane. It doesn't have cytoplasm. It doesn't have membrane-bound organelles or organelles at all. And it doesn't have ribosomes, which means it can't make its own protein either. It's not able to maintain homeostasis or reproduce. So, because of that, it's not a living thing. But it does have genetic material and it can evolve, which is something that living things do all the time. So how do we know about viruses? Well, there's a part of science called virology. This is the study of viruses, and virologists are people who actually study specific viruses and how they spread through a population. We have a group of people that just studies AIDS and chickenpox and the rhinovirus, which is the common cold, and the coronavirus group, which is also common cold, and influenza, which is our seasonal flu. These people have developed vaccinations or vaccines that allow for us to produce immunity. Not all viruses create a huge response in us, so we don't have vaccines for every virus that's out there. And some of them we haven't been able to develop a vaccine for, such as AIDS and HIV. So viruses, where are viruses at on the size scale? So here's what we have. Viruses are much smaller than prokaryotes. So this is an Escherichia coli down here. This is a prokaryote. And up here we have a red blood cell. That's enormous, by the way. And then all of this stuff right here, these are all viruses. This is rabies, Ebola. Here's a bacteriophage, which only infects bacteria. Another bacteriophage. Here's a rhinovirus. When we're talking about small, think about this. That's a rhinovirus right there. So when we start looking at some of these, they're really tiny. They must have a host to replicate their genetic material, and they can be found anywhere that life is found. We are going to talk about phages. These are specifically useful viruses, which only infect bacteria. We use them continuously in research. So let's talk about the structure of these viruses. So general structure, a virus is a particle that consists of DNA or an RNA within the center, protected by a capsid or coating. This capsid is a protein that allows for it to gain some protection against the host's immune system. Remember, they're trying to invade a cell and infect a cell, so it kind of is camouflaged. Our own body is camouflaging these viruses inside of us. But it makes it a lot easier because of this coating or this capsid to infect the host cell. And so here's some shapes. One virus is a helical virus. Look at that little swirl right here inside. So this is the helical DNA. It's in this hollow core of the capsid. And that is in the center. That's where all the genetic information is. It's typically rod-shaped or filament shaped. And right here, this is an example of one. This is a tobacco moss virus. This affects tobacco plants. P.S. And by the way, one of the first viruses that was ever studied was this virus. Importantly, because it was destroying the cash crop known as tobacco, and we were losing millions of dollars. Okay, yeah. So a huge economic crisis did cause us to look into this virus and so many other viruses as we go through this. ICOSAHEDRAL viruses, these are spherical in appearance under low magnification, but they really have a cool, complex genetic pattern, and so they're not truly spherical. But we can see this is the capsid. Inside of the capsid, which we cannot see, would that be that genetic material? So they have a single basic unit of protein, which is just repeating and repeating and repeating. And so it can save space within the virus's genome. And so it's really a very small portion of the genome actually codes for the capsid, and the rest of it codes for the DNA. And then we have complex viruses, such as our bacteriophage here. Bacteriophages are really cool because there's multiple parts to them, and that's why they're complex. They have a head, which is consistent of the capsid with DNA inside of it. Then they have a neck and a collar with a sheath on it. Then they'll have a tail and a base plate. Basically, what this does is it acts as a huge syringe. It lands like a spaceship onto a bacteria, and then it throws a syringe in, and then it shoots all the DNA, and then this whole thing goes away. Then we have enveloped viruses. These are viruses that surround themselves in a position in the host membrane. So they actually, here's the virus right here, and then it comes in contact with your membrane, and it surrounds itself with your membrane. It literally is camouflaging itself with your own membrane. That is crazy. It can do it not only from like your cell membrane, but from your membrane of your Golgi apparatus, from your nucleus. It's crazy. Anything that's membraned within our cell, it can adapt and hide itself there. Example of that, HIV. That's why it's so hard to cure. We can't have a vaccine for HIV because we don't even know how to like get it out because it lays dormant and hides within our own membranes. Influenza is another example of this. This is a huge advantage for viruses because the host immune system can't attack it because it's being disguised literally by its own immune system. And so it becomes part of the cell, and so it doesn't have any identification of the actual virus. It's just your membrane. And we don't want to kill our own stuff. So let's talk about how we are classifying these. So viruses are classified based on their capsid shape, either DNA or RNA, type of nucleic acid, the host organism in which it infects, the type of disease it caused, and the presence of an envelope or lack of envelope. And so when we see this, we have like HIV, the human immune deficiency virus, which does have an envelope, which hides in our cell, a bacteriophage, which has a complex structure, but does not affect us because we aren't a bacteria. An influenza virus. Influenza is seasonal and super changing. Again, it adapts because it has an envelope. Adrenoviruses are tobacco virus, and then the hemorrhagics, Ebola. This disease, thank God, does not spread easily. Otherwise, we could wipe out a lot of the population because it has a pretty high mortality rate. Okay, so now this kind of breaks all of that down and classifies some of this. Now, here's the one I want to talk about. We have a complex capsid. It has RNA. It creates a common cold acute respiratory syndrome known as SARS. And look at it. It's a coronavirus. Coronaviruses aren't new. There's something that's been around for a long time. One of the problems with it is it does have an envelope, so it masks itself. So as we start talking about some of this stuff, it's a lot harder to create these vaccines when they can mask themselves so easily within the human body. So let's talk about the origin of viruses. So there are three main ways that they think viruses have gone. So there's a regression theory that proposed that viruses arise from free living organisms that became intercellular parasites to depend on the host and loss of function. So basically the virus was just this parasite that went into the host, and then it just did so well in the host. It went, the host could do everything for me, and then it lost all parts of itself that were maintaining homeostasis. Then there's the runaway pieces theory. It's kind of like the prion theory. We'll talk about prions in just a second. So basically, a runaway or rogue piece of nucleic acid broke off and then started living independently from the host cell. But it was just a piece of nucleic acid, so it couldn't reproduce on its own. And then it could infect another organism or cell and then make its DNA and actually a little host. So the runaway piece theory is also what we think about prions. Prions are a protein that becomes altered. A prion disease is incurable and it's fatal. And so mad cow disease would be a prion disease. As soon as you come in contact with that protein, it just spreads like wildfire through a population. And so you can look up mad cow disease if you want to see some prions. We also think that viruses could have had coevolution. They've always been around. They were here. They're the most primitive thing that we've ever seen on this planet, and they kind of just lost their self-replicating abilities because, well, they could infect so well, and so they didn't need them. So that's one reason too. So how do viruses replicate? So we said we have some different nucleic acids. So let's talk about RNA viruses. Some of the RNA viruses will directly affect, so they'll take the viral genome and put it in so that it assumes, or our cells will assume that it's the mRNA, and it'll just translate it. It's crazy, right? And it just starts translating, and we just make whatever in translation we're told to make. Some will carry enzymes that will allow for the viral RNA genome to attach to the actual host's genome. And then we go through a viral mRNA, and it will just keep reproducing through transcription and translation that way. Retroviruses, these guys are crazy, by the way. They actually take the viral DNA and integrate it through reverse transcription into the host DNA. So it's always there. So when it splits apart, so like when DNA helicase splits the DNA apart, then it's actually replacing it with viral DNA to make the complementary strand of the host DNA. So then it's always going to be in the host DNA for that cell. And when that cell replicates, it replicates the viral DNA too, which can be problematic. DNA viruses are a little different. So here's our little virus here. It goes in. It actually will integrate itself with the nucleus of the host cell, and it'll actually piece its DNA in with the host DNA. And so this is really cool. It's done through a process called reverse transcription, which becomes very useful for genetic engineering later on. But basically what the host will do then is when it goes through and it reads that part, it just makes a new virus, and that new virus can go out and free float into the world. Bacteriophages, we said, go through 2 phases, and bacteriophages only infect bacteria. So one of them is the lytic cycle. It's where the bacteria will first attach itself to the bacteria. So the bacteriophage attaches itself. It plunges its DNA in through penetration, and then goes through biosynthesis. So the host cell will read the phage's DNA, and it will just start making all the proteins it's told to. And then after it's completely matured and it has fully assembled all the particles here, so that we have to assemble them, then it will lyse the cell. And basically the cell breaks open and bacteriophages spray out everywhere, which is kind of gross. The other thing is the lysogenic cycle. The phages will infect a host cell. Now, it's kind of the same way. It lands over here, it throws its DNA in, and then what happens is this. It incorporates the phage DNA into the bacterial DNA or the plasmid. And so if it's not stressed, that bacterial phage DNA just kind of hangs out and gets replicated and replicated and replicated and replicated. But when it is stressed out and it's under really stressful conditions, the bacteriophage goes, oh, this host, it's awful, let's get rid of it. And it goes right into the lactic cycle. And so then the bacteriophage DNA will replicate and make a ton of bacteriophages until there's so many assembled that the cells lyse and the bacteriophages go out. And it'll start this process over again. Bacteriophages are really kind of important, and we'll talk about them when we do research here. So human diseases. So I have a list of human diseases that we have over on the left side. Viruses, I want you to know, have latency, which is they can lie dormant within your body. HIV and AIDS are those type of viruses. We can have an epidemic, such as a seasonal epidemic, such as, it's called influenza. That's why you get a flu shot every year. These are outbreaks of disease within a population. It can happen at a high rate. These epidemics are pretty seasonal. Like, we know we're going to have an influenza epidemic every single year. However, a pandemic's a little different. This is where you have an outbreak of disease over a large area, aka the world. Influenza has had a pandemic. We're in a pandemic right now with the coronavirus. COVID-19 is basically looking at the coronal infectious disease, and it started in 2019, so that's why it's got the 19 with it. This is a new emerging disease that has been transported much faster than we actually thought. And we know that it's gone through zoonosis, which means it's spread from animal to human. When a disease does that, that's usually when we're going to get an emergence of this. The avian flu, SARS epidemic, COVID-19, these are all examples of this. One thing that we need to remember too when we're talking about these, viruses can live inside of us for a very long period of time, dormant, and then they can be reignited for some reason, especially if we have immunocompromised diseases. HIV would be one. When we talk about chickenpox, if you've had chickenpox, you can get shingles when you're older. And so they do lie dormant, and some of them may never appear and some of them do. Herpes simplex 1 and simplex 2 are examples of that. You get a cold sore, and then you'll be years before you get the next one. So they can hang around for a long time. But how do we control these viruses? Well, we do so with inoculations and vaccines. So they're a little bit different. An inoculation is where you actually take a small amount of the actual virus. It can be heat fixed or alive, and you put it inside of a human. Scary, right? We did that with smallpox, which is a disease we've kind of tried to eradicate. We also have vaccines, which are a mixture of some anti-genetic material and other immune stimuluses that allow for an immune response, such as we use cowpox, which is like a weakened type of pox virus, which we could treat or build immunity with smallpox. No, vaccines that are not inoculation vaccines, these ones have to be repeated. So you'll have to have boosters throughout your life. Otherwise, like if you get pertussis or whooping cough when you're older, it's probably because you haven't been vaccinated for it. Like you should get a tetanus shot like every 7 years. We also have some antiviral drugs. These are really common for people that have infections like HIV and AIDS. These are medications that are used specifically to treat the symptoms of a viral infection. They cannot cure the viral infection because the virus is incorporating itself into your cells and your DNA, and your cells have to die in order to kill the virus. So antivirals are simply to treat the symptoms so that your immune system can fight the actual virus. We don't have any drugs or medications that can cure a viral infection. So if you're going to the doctor because you have the flu and he's telling you, hey, you just need to go home and get some rest and drink lots of fluids, and they're not giving you medication, it's because there is none. Tamiflu will just help with if you have high fevers, diarrhea, that kind of stuff. But if you don't need to take it, don't take it. So viruses are really important for research. And here's why. We talked about those bacteriophages, right? So bacteriophages allow us to better understand transcription and translation. Immunology, we can trace the viral DNA easier than we can trace our own DNA. We've actually used bacteriophages as tools to replace sections of genetic material so that we can create new things in a petri dish. An example of that, in genetic research, we can use a technology called CRISPR, and we've actually developed that from bacteriophages. So we can just take those segments of DNA and piece them together now. And so genetic engineering is based on viral research. We can also use viruses for gene therapies, which sound kind of weird. So we're trying to research how we can have them make proteins for us, so we can insert a segment of DNA that may be missing, and then it can create a protein response in there. And so there's a lot of research that viruses have allowed us to understand the inner